Yeah. I mean, I love this. It's great to see that the room can contain the crowd. Yeah. Well, I guess, yeah, we can sit also on the floor if you don't mind. I'm sorry. Yeah. All right. Welcome, people. I can see that there is the seat taken? Oh, okay. There's someone's bag on a seat. So, okay. Thank you, everyone, for joining. We're going to start at the hour, but very happy to see that there is interest and then people are coming and we have more people than seats. So that's nice. I'm going to start just with just a little title slides to introduce the session. My name is Marie Grosjean. I'm the session chair, and we have Laura Russell, who's the session co-chair and co-moderator online. And thank you, and welcome to our session that's titled Exploring and Enhancing Collection Metadata with the Global Registry of Scientific Collections, also called GRSciColl. Yes, we have to thank our diamond sponsor for and the Okinawa Convention Center for today's tech support. Thank you very much. So yeah, our diamond sponsors are Picture, Delta Designs, and Max. There's going to be about presentations for 10 minutes. You don't have to read all of that. That's just so that I remember that it's there. But the session is recorded. We'll have 10 minutes presentations, and we also have time at the end of the session for questions and more hands-on work. So if you want to stay afterwards and try some things yourself with GRSciColl, then you're very welcome to do so. We are really a lot of us here today, and we can help you now or even later during the conference. So don't hesitate. Thank you. And now we're going to start with Melissa's presentation, who's going to introduce us to GRSciColl. Melissa.
Hi, good morning. Oh, I feel my voice is better. I'm Melissa Liu from the GBIF Asia Regional Support Team, and welcome to the GRSciColl session again. I'm the first one, and I will start from the basic introduction of GRSciColl, but unfortunately, as you can hear, my voice is a bit cracked today, so yeah, don't be shocked if I present in a weird way, okay? And okay, GRSciColl stands for the Global Registry of Scientific Collection, and it is a website and a catalog contain the information about physical scientific collections and institutions. And it's also contain the collection and institution codes and identifier, which are also used to link specimen and literature-based occurrence to publish on GBIF to GRSciColl entrance. So it is also community curated, and you will know more in details in this session. Okay, the next presentation, you will know how to get involved in it. And it is also noted that GRSciColl has an API to access and edit content programmatically, and the content can be integrated in hosted portals like the Disco UK and Disco Flanders portal. And by the end of July, GRSciColl has connected to more than 8,500 institutions, includes almost 8,000 collections across the 252 countries. And we also have 54 mediators responsible for 114 countries. And how comes the numbers on GRSciColl? Once the collection data is published in GBIF using the same codes or identifiers for institution and collections as is registered under the GRSciColl, the specimen in GBIF will be like automatically linked to the GRSciColl entrance. So who and what is GRSciColl for? For the community, they can find collections and contacts for collections of interest. And for institution, they can make their collections like more discoverable, visible, and showcase their own collections. And for external systems, like they can improve system interoperable and enables the data linkage. And for like national organizations such as like GBIF nodes or others or governments, they can help guide the data mobilization and facilitate the monitoring. So what information you can access from the GRSciColl and I will like introduce the basic features on GRSciColl interface. The information for users to access includes the collection location and content, accessibility status, and coverage, for example, geographic and taxonomic information, and also the context of the museum institutions, and also images. And there are like 3 ways to search data on GRSciColl. You can search by the institution and collections or specimens. And let's like take the institution search for example. And here you can also like search in free text you want or filter the country, area, or code, or the numbers of specimens. And we take Japan, for examples. If you filter Japan here, you can see all the institutions, collections, and in Japan as the map on the right side, and see all the results related here. And if we click one of each result, let me see, okay. You can see the description, like includes the institution code, numbers of specimens, records on GBIF, or like founding date, institution types, other information of the institution, and also the contact information and the identifiers. And also we have the feature we call related, and it can like automatically detect records they may be related. So the examples could be like a specimen sequenced by another institution, type material deposited at different institutions, or 2 people like reporting the same bird at the same location and time. So the view like shows the occurrences from the result, and for each like records shows related records together. And the same cluster may appear multiple times, as it is not a list of the distinct clusters. So, but of the occurrences, the entry point for each cluster is like highlighted, so you can see different group of the clustering. Okay. And in collection search, we also like the filters are also like similar, and the result will also show the numbers of specimen and the numbers of the specimen records on GBIF. And also we take Japan for examples, and you click if you click the results, and you can see, you will see the description also, and the map on it, and the top 10 taxa, and also the collection descriptors. So let's go a bit deeper. Let me introduce about what is the collection descriptors. So until like recently in GRSciColl, most of the description fields for collection were only like free text fields. For example, taxonomic coverage. And these are quite like, difficult to search as they can contain many different things like birds, aves, and so on. And some collections are like able to digitize their specimen and share the records on GBIF. These can be like indexed and searched easily. So collection descriptors are like for everything in between. For example, a collection can like share a list of higher taxa that they have in their collections. Thus, like making collection discoverable when like someone is looking for a taxa described in the list. So collection descriptors like should be mapped to standards like Darwin Core or Latimer Core, and whenever possible. So, however, they can like also be used to share additional information like which cannot be mapped to Darwin Core or Latimer Core. So unmapped data cannot like be indexed and searched though. So this is like how the descriptors work. And for the collection part, that like the descriptor, we can like see, take a look for the example. We take the most in Japan, for example. So you can see the collection descriptors in the page. And if you click the preview button, it will like show some text in detail and that provided by descriptors such as like individual count and the number in database and number of imaged collections, and they can be like also downloaded. So this can be useful for collections that they are not yet digitized, for collections that have specific characteristics that are not covered by the standard collection metadata. Okay, and there is a new feature we call like dashboard. It can like show based on the share, based on the records shared via GBIF. So it includes like statistics, data richness, and IUCN threat status. Okay, and we will like see more deeper in the dashboard part, but it's almost, the visualization. And okay, and if you like go to see the dashboard and do the filter, we take the Orca and Mola Mola here, for example, and search some like specimen species. Like you can customize the metrics of the species on the page, and like the tables here, and or map or gallery, and then create new metrics, as many as you need. Okay. So if you want to like know more in details and know how to get involved as the descriptors or like mediator or editors, like please stay for Chihjen's presentation later. Okay, thank you.
Thank you, Melissa. We don't have time for question right now, but if you can stay until the end of the question, and if you can use sign language, Melissa will answer without using her voice, because I think we've exhausted it now. Please, yeah, there is space to sit on the floor if you want in the front. And I can see a chair that's like empty right there if someone wants, and the one here, yeah. So for the next speaker, we're gonna have Chihjen. He's gonna explain to us how you can edit. Isn't it Chihjen? Oh, sorry, Lily. Sorry, my bad. I'm a great chair. Believe me, I've done that many times. Sorry, Lily. I got thrown off by Melissa's last slide. Lily is gonna tell us a little bit more about how she and many other GBIF contractors are using GRSciColl to mobilize data to GBIF. Thank you, Lily.
Thank you, Mary. Hello, everyone. Good morning. My name is Lily Shrestha, and I'm from Nepal. I'm also one of the member of GVIF Asia Regional Support Team, together with my colleague Melissa, Chihjen and Vijay here. Today my presentation is entitled Initiatives to the Global Representation of Scientific Collection. I will directly go into the presentation because you have heard about GRSciColl just now from our colleague Melissa. In GRSciColl, most of the institutions, more than half of it, are mostly from Europe and North America, and there is very. That was my bad.
We couldn't see Lily's face.
Oh,
okay. Sorry.
I'm making
Lily,
sure that.
Please continue. Once again, I'm great, Sharon. You can hire me anytime. Stress testing.
So as I was saying, there is very less representation in GRSciColl from the collection institution from other parts of the world except Europe and North America. More than half of it is from that region. So we are making an effort in GVIF to improve the access to the collection information from the underrepresented regions of the world. So GVIF is doing that basically through GVIF's participant network and its regional support teams. GVIF's participant network have always been actively encouraging collection institutions from their region or area to share the data both through GVIF and GRSciColl. And we, the regional support teams from GVIF that are from Asia, Africa, Latin America, and the Caribbean, and Europe, we are trying to complement the effort from the participant network by updating and adding entries in GRSciColl, while also trying to support any data mobilization activities that we can in those institutions. So for this work, we have a definite workflow that we follow, which starts with us trying to update the entries of institutions and their collection by verifying their information through, for example, their web pages or any other online sources that is available. The next thing that we do is check whether they have already published their data in GVIF or not. And as a third step, we try to contact those institutions, first of all, inviting them to update, suggest the update to their institutional entries or collection entries. And we also try to inquire about any of their plans in mobilizing their collections through GVIF. And if they are interested, we try to offer technical support with their any publishing related, GVIF publishing related activities. So to keep the track of the work that we are doing, GVIF has maintained a GitHub repository that is called Collection Mobilization. As you can see, the issues in that GitHub repository represents to corresponding institution in GRSciColl. And what we do here is, after making the changes in respective institutional or collection page, we try to provide the comments, as you can see down there, mentioning about the changes that we made in GRSciColl page. And we also try to give them labels, identifying whether they are already a GVIF publisher or not, whether they have published any data set in GVIF or not, whether they have been contacted, or whether they have agreed to mobilize their data in GVIF or not. And if they agree to mobilize the data, as in the case with this institution from Singapore, we add their data set in another GitHub repository that is called the Data Mobilization Repository, as you can see here, and we keep the track of data publishing process through that. So we basically not only update GRSciColl entries, but also encourage the institutions to mobilize their collection data through GVIF. This is the progress of all the regional support teams so far. So out of total 2,449 institutional entries, we have updated 458 institutions, and other 592 updates are in progress. We also found that some of the entries were duplicates, so we tried to merge and delete 82 of them. We have established contact with 88 of the institutions, and out of that, 19 have expressed and are in the process of mobilizing their collections. And so far, out of this work, we have successfully mobilized more than 161,000 specimens. So the next part of my presentation, I will be taking you through the cases of data mobilization from GRSciColl institutions. I'd like to start with the institution that I personally was involved with. That's called the Natural History Museum in Nepal. So I basically contacted the institution to see if they were interested to mobilize their collection, and they requested a training support to understand the process of GWIF data publishing. So I conducted an in-house training for their staffs and interns about digitization and the process of data publishing in GWIF. And they started digitizing, and they have published so far 9 data sets containing 7,294 specimens, and few other data sets are in progress. This map here in the right shows the specimen distribution. So you can see it is all from Nepal, and that are the data sets published so far from the institutions. Next is the National Museum of Natural History from Bulgaria, where our colleague Salza works in. So with her technical support, they have also mobilized 2 herbarium collection on vascular plant and lichens with around 7,392 specimen occurrences. And the digitization is ongoing in the institution for 28 of the rest 30 collections. So these are the data sets that has been published. Again, the map on specimen distribution and the date of collection for those specimen. So I'm not going to describe the slide because I have limited time. And the next case is again from Bulgaria, again with the help of our colleague Salza from the regional support team in Central Asia and Europe and Central Asia. So with her technical support, this institution, Sofia University, have published their geological collection in GWIF with around 6,716 specimen occurrences. This is the specimen distribution, all from Bulgaria, and the collection date for the specimens that has been shared in GWIF. Next are the 2 institutions, the coming 2 slides that our colleague Leo was involved with. The first one is called Instituto de Biodiversidad de Venezuela. I'm sorry, I'm not right. I may not be right with the pronunciation, so I'll just go with INBIO. So with his technical support, they have mobilized their herbarium and fish collection with around more than 78,000 occurrences specimen, and these are the data sets. The specimen distribution, mostly from Venezuela and other parts of South America, and the collection date for the specimens that are published. Again, another institution, I'm not going to pronounce this because I can't. So IVIC from Venezuela again. It is one of the Venezuela's 4 largest herbarium. And with the technical support from Leo again, they published the herbarium collection in GWIF with, you can see, a lot of specimen occurrences, more than around 62,000 specimen occurrences. Again, mostly the occurrences are from Venezuela and other parts of South America, and the collection date for the specimens that were published. And as a last case, we have this institution from Latvia that our colleague Oleg was involved with, Daugavpils University. So with his technical support, they have mobilized one of their zoological collection and 1 botanical collection with around more than 70,000 specimen occurrences. And the digitization is ongoing for rest of their 2 collections. Specimen distributions are mostly all from Latvia, and the collection date is represented in that graph. So this was about the collection mobilization efforts that we have made so far, and we are in the progress of mobilizing and contacting more institutions and trying to mobilize their data in GWIF. However, we have encountered some challenges in the process, so I'd like to highlight some of the challenges. So when we are trying to update the collection institution entries, sometimes their web pages are simply non-existent, sometimes inaccessible. And even if we can sometimes access the web page, there is very or no information about their collection that they host. So sometimes there's no information about the contact as well. So we don't know who to contact to try and get their information updated. And even when there are contacts, sometimes what happens is when we are trying to establish contact with the institutions, we simply do not hear back from them. So that's what is making our effort of this work a little bit difficult. While despite the challenges, we see a lot of opportunities with this work. So we are continually trying to improve and expand the information in GRSciColl and also improve the access to collection data through GWIF. So through this work, people will be able to have an access to updated data on institution and their collection. They can also have access to some of the missing collections that were not originally in GRSciColl. They can have information on updated contact information, and most important of all, we are trying to get as much data from the collection institution as possible in GWIF. So more collection data will be available for open access and for use. Thank you very much for listening to my presentation. I would be very much happy to answer all your queries with all my colleagues. Thank you.
Thank you, Lily. That was great. And thank you, and you, all the contractors, for all the hard work you're doing. It's not easy. Okay, so once again, I think we'll have to skip the time for question for now, but keep in mind that we'll have time after the talks. So now the next talk is from Sharon, and she's going to tell us a little bit more about Latimer Core and GRSciColl. Great to see everyone here. There is a spot here on the side if you want. Oh, it's taken. Okay, yeah. Oh. Once again, I treat my speakers well, I promise. You all will be invited for drinks later, speakers.
Yeah, actually. We love you, Marie. Really, we do. Cool. So I'm Sharon Grant from the Field Museum of Natural History over in Chicago in the United States. Quick question. How many of you were in the Latimer Core session earlier this morning? Hand up. Okay, good. So I'm not repeating myself to too many people. Okay, so I'm here just to talk through the Latimer Core standard, which was ratified yesterday, yay, and how it's intersecting with GRSciColl. So let's move through this. So I'm going to go through 6 things real quick. We'll talk about Latimer Core and where it is right now. We'll talk about some of its basics and how it hangs together. You've heard all about GRSciColl, but I'm just going to quickly flick through some stuff to connect the dots. And then I have 2 challenges that have come about through the implementation of Latimer Core as part of GRSciColl. And at the end, hopefully, maybe you guys can help me work through some of those. So I'll look at those with you. Okay, so Latimer Core. Status update. There was a presentation on Monday, was it Monday? And they had a big slide and it said that Latimer Core got ratified. We went through the public review phase back in December through January. We got the experts to come back and tell us what to do, and we are now ratified. So there are some links here to the normative parts of the standard. And what will happen now for that task group is we go into maintenance mode. So rather than meeting once a week, as we were through the pandemic, we'll go to monthly. So if anyone's interested and you want to jump in and ask questions, have a look at our wiki. It'll tell you when we're meeting and what we're doing next. So again, just through our documentation, we have a wiki. We use the GitHub wiki a lot. And if you're new to the standard and you just want a quick, what does it mean? What does it do? What did Sharon say? I can't remember. The wiki, go to the wiki page. If you don't use GitHub a lot, go to the wiki page. Just find the wiki in the top. And we've tried really hard to write it in Humanese. So it's English Humanese. If anyone would like to help us translate it into other languages, we would love you to do that. So unfortunately, it's in English Humanese for now. There's also the TADWIG page, and then the normative page. So if you're looking for the actual terms and the definitions, there is a page. It has a purple header, and it's the equivalent of that Darwin core page where you have the terms. Right? So that's your quick guide to the bits and pieces. So basics of Latimer Core. What is it? Well, just to give you a bit of a heads up, I know we don't name things after people anymore. So technically, we named this after the coelacanth, which is a fish that was known to a lot of local people, but the scientific world had forgotten about it. And so Latimer Core is really about trying to give people an easy way to talk about their collections. Everyone in the collection knows about the collection. We want you to be able to talk about it to the broader audience without having to database every single specimen, because if we could have done that, we'd have done it by now. So that's really its overarching purpose. Who's it for? So there are 3 groups of people. And they use the same standard in slightly different ways. So we've worked through the situations for aggregators, data providers, and then for the people at the other end, the practitioners, the people who are using it. So we've really tried to give 3 views to the standard. And we'll talk a little bit about how those play out. So there are two core concepts to Latimer core. The object group and the organizational unit. So you've seen from GRSciColl that we can now start to put these lists of institutions together, right? And we can say, you know, what was it, when was it founded? What's its address? What's its general scope? What type of institution it is? Is it a university with a museum underneath it? Because there's also, you know, each institution has these different levels. And then allied with that institutional unit and its hierarchy are the object groups, the things. You'll find that we don't use the word collection a lot because collection has all sorts of connotations around it. You talk about the collection as the part of the institution, you talk about the physical collection. So we use object group. You'll see object group as a term. And what you can then do is, to any of those organizational unit levels, you can assign object groups. You can say that the biology department has a botany collection, or you can say that the field museum has a botany department and it has a botany collection. So it's for you to be able to define your hierarchy rather than having somebody else impose that hierarchy on you because they designed it first. So those are the 2 core concepts for you to get your head around. So like any good standard, we have a bunch of classes. We have 25 at ratification, and we've tried really hard not to recreate the wheel. So hopefully if you've used Darwin Core, if you've used ABCD, you should recognize some of these classes because we're describing the same things that they are. We're just not doing it at the specimen level. And only where we really, really couldn't find something from elsewhere did we create our own, hence the organizational unit, the object group, they belong to Latimer core. We only have 3 required classes, or mandatory classes. A thing called Latimer core scheme, the object group, because you're talking about collections, so you have to have an object group, and a thing called record level. And I won't dig into the details. Read the wiki, it's all in there. But essentially what you're doing with the Latimer core schema is you're defining the shape of the standard for your implementation so that anyone that you give that to can understand the data that you've asked for. So rather than having 16 different fields that you get that request for, now you can have a standard shape. And the record level is the thing that has the licensing around. So let's talk about the what we call the scheme classes. Like I said, I just talked about the Latimer core scheme a little bit. I'm going to focus a little bit on the scheme measurement of fact, because not only do you want to say, I am the field museum, or I am this institution, you want to say what you have. You want to talk about it and give statistics and metrics about it. So we use this thing called scheme measurement of fact. If you're a Darwin core, we borrowed it from them, because why reinvent that? One thing we did add to it, though, is we added, let me just get this right, measurement fact text. If you know Darwin core, you have a value. But that doesn't let you, if you then mix text values with numbers, you can't add anything up. So if you want to know how many plants we have at the field museum, right? If I've written the number 2, or you can't add that to something else, right? So we've split numbers from text. And as you can see in this example, you give it a type, object count, what's the value? That's a big number, 44 million, I think that is. And if you want to talk about your collection history, you use measurement fact text, right? And there's your text. Like it says here, so you use values for quantitative things, and if you want to describe things, you use the other partner. So fact text belongs to Latimer, please feel free to use it. The other one came from Darwin-Core. Again, for people who don't know these things very well, classes have properties, and a property is just the detail, right? And each property has its page on the norms. So, and then within that, the attributes are the actual things that you, the actual data. So in this case, you've got the class taxon, the properties genus, and then you've got the attributes. We also embed, unlike Darwin-Core, we embed relationships and repeatability. So we give you some hints on how to connect things up. I'm going to not spend a lot of time on GRSciColl because you heard all of that, but there's the cute little graphic. So the collection descriptors, you saw those earlier. That's the real connection point between Latimer Core and GRSciColl. So I have two shared challenges. Control vocabularies. There was a whole session on vocabularies yesterday, and this is an issue. Latimer Core, we give very few mandatory hard, you must use this control vocabulary. That we want to come with the schema. So when you, as the aggregator or the request of the data, say, this is my schema, you should define your vocabularies for people to use. But what's the best way to do that? So that's my challenge to the group. How do we do that well and simply? And then the other one is around metrics. MIDS is out there in the world. And the challenge we have is, can we use MIDS at the collection description level? We can certainly use it at the specimen level, but is it appropriate at the collection level? So those are my two questions to the group for later. And thank you, Marie. And thank you to all the people who worked on Latimer Core. We got it across the finishing line, and to the people who came before. It took a while. So.
Thank you, Sharon. Yeah, lots of challenges. And actually, for those of you who are interested, please stay afterwards, because we, once again, don't really have a lot of time to discuss right now, but it would be really nice to have your input on this. All right. This time, yes. Speakers know better. We're going to listen to Chihjen, and he's going to let us know a little bit about how you, or people you might know, would want to edit information in GRSciColl. Thank you.
Hello, my name is Chihjen Ko, one of the Asia Regional Support Team. My part of presentation is very practical, so I'm going to ask you to sort of take a breath and slow down. And what I'm going to do is take you a visual journey on the editing interface on the GRSciColl. So you're going to need to put a little bit of attention to the website screenshot on the screen. We are inviting communities to contribute contents on the GRSciColl. As you heard from Lily earlier, that it's not always we get response, even we want to contribute to update information on the specific institutions. Therefore, it's an ask to the, all of you who are involved in this business, and but there's got to be some incentives that when you put your editing efforts in the GRSciColl, what you get is getting your institution to be visible and findable. And for people who want to find collections and find the right person to talk about specimen long, for example, and they can quickly go to the right person, because not many museums have the budget to actually maintain a well indexed Google searchable website to keep the communication channel smooth and frictionless. So it's an effort that a little editing efforts you put there, and they will be rewarding afterwards. There are majority two types of entries on GRSciColl. One is institution, the other one is collection. You can go to edit and suggest institution information directly, and also as well as collection. And the editor can do create, update, merge, and delete of these entries. Specifically for edits, you have to have a permission, of course. So what you need to do is to have an account. And but if you don't have an account, you can always provide your edit and updates, and that would be sent in as a form of suggestions. And people with the editing and mediator right in the back, they can review, change, or double check with you, or even email with you to communicate a confirmation about the detail you submitted, and once it's this thing is legit, and they will apply the suggestions. So when you go to the GRSciColl website, look for first on the top part. There's the how-to, and when you click that, you have add entries or edit entries. Here, I want to invite you to go first, add entries. Then you will be leading to this page. There you can add new institutions or new collections. That's when you so far couldn't find your existence on the GRSciColl platform. But I'm showing the form that is also applicable for some editing activities afterwards. So in this case, when you just go there, you don't have, you are not logged in as an editor. So it will explain to you that since you don't have edit access, you can suggest change. And that is a must. At the end of the form, you provide the email. And the top part, as you can see, there are several very basic fields that you can put in. Particularly important is the code. That's usually the institution code. Usually the code that in the monograph, manuscript that when a specimen cites the institution code that is used on the literature. And there are a bunch of type additional code that you can put there because among the literature maybe code changes. And when you think it is necessary to be there in order to provide people references of possible alternative of codes there, you're always welcome to put extra codes there. And also indicate whether your institution, the institution is active or not active. Of course, if it's good if you have one web page, then so people can go to your website. How to become the editor? When you click that, you go to the part that gives you a detailed explanation of what you need to do to become an editor. Usually, we'll consider that if the email you send is associated with the institution, and we will give you the editor right of that institution. And you need to send that email to scientificcollections@gp.org. So as the form goes, this is the first part. And the second part, also you can put in email, catalog catalog URL if you already provide the, this is for the case that you already have a website for people to browse your collections. And if you put a latitude, longitude, it will show exactly where your institution located on the map. In the later part in the form, we are trying to do some address mapping. So if you put address there, and then the system in back will try to analyze the address, probably use the Google services. Yes, experimental. So, but the best way is that if you look for yourself and address the coordinates on the Google map, you put in the coordinates in the earlier part, and it will show correctly where the map is. And also, you have the opportunity to decide that on the whether you want your institution to appear on the scientific-collections.gp.org website by clicking this checkbox. It says that display on the NHC, and NHC stands for Natural History Collections Portal. And if you click on this, it will be visible on these. Sometimes you want to just put some start to provide your institutional information, but if you're not sure that you're ready to expose in the wider, so that's an option you can choose. At the end, it's very important that when you suggest this institution information, you provide your some comments and your email, and we will reach out to you. Second, I want to use a example of editing collections, so you can have a look at what are the fields available on the collections form. And in this example, I first go into look for a collections in Thailand. First here, collections, and filter by Thailand, and I find this stock of insects specimens that's interesting to me. And I click that, and then I find there's some information here I want to update. So I click Edit, and it will bring you again to the backend editing page. And if you don't have the right to actually edit that, again, you have this suggestion button you can click on, and it will bring on the form as if you are editing that. So we're going through that, what's available there. And this is the second part. Again, that's the relevant fields that you can quickly look on the left part. That are the things you can provide for describe the collections information. Okay, that's the example I put there, and click Save. In terms of merging, if you need to associate these collections into a institution, it's not merging, it's simply to associate a collection with an institution. What you do is in the institution column, what you can do first is to try to search the institution name where this collection is affiliated. But another more precise and secure way is to find out the UID. And the web interface will try to interpret the UID and find exactly the collection, because there are many institutions that have similar names in the string. So the automatic search probably won't very quickly get you to find the one you actually want to affiliate in with. How to find the UID? So on the specimen, on the collection page, click the link to the institution, and on the top of the URL, there's UID on the URL. So you can just copy paste the UID and you get that. Okay. Finally, I think the key part is the how to successfully link the specimen with your GRSciColl institution. GBIF tried to use the code to match the specimen that is already published on gbif.org. But here I want to invite you to take a look at the identifier here of the institution. It's very small, but it's fine. So a more secure way is that when the specimen information is published, use the identifier as the institution ID. So as soon as the specimen is published, that will be a precise association, just to guarantee that the specimen record is correctly mapped to the institution. So here is an example of Royal Belgian Institute of Natural Sciences. When you click the specimen record share with GBIF, the specimen number, you go to the specimen page, and when you click one of the specimens, you can see that the institution ID is using the string that I showed in the previous web page. So I don't want to go to descriptors because I see Marie is a bit of, worry about the time. Descriptor provided an option for you to upload any structured information to use to describe your collections. And that's what you get when you download. It's whatever the structured format, and it doesn't really matter about the columns definitions. Of course, later there will be sort of ongoing development synergy with Latimer core, so people use that to describe your put it in the description. And right now, you can already see that for those who has put a descriptor use the descriptor features, you can already see that in the GRSciColl website, and you can preview that or also download the part. So what I want to end is because this is supposed to be connecting to hands-on exercise to use that, and we try to deal with any features or any questions you have when editing. But I guess what I want to say here is that actually the development of this system probably comes from your tax paying to your government. So make use of it, please.
Thank you, Chihjen. So if you're interested in trying uploading collection descriptors in GRSciColl, Chihjen is here to help you. I can also help. But it's like mentioned multiple times, it's a pretty new feature. So yeah, we don't have extensive tutorials yet, but we're here. We're on site. We can do this. We can buy you coffee and help you through this. Well, I can buy you coffee. I cannot promise for the others, but... Yes, exactly. Laura is reminding me that GB folks, we have a session of help desk in the place where the booths are. You can come to us during the last time slot of the day, every day. We still have 2 days left, and you can show up and ask questions, and we can help you with not just GRSciColl but any GBIF related questions. I'm just thinking any GBIF technically questions. Okay, so thank you very much, Chihjen. Once again, we'll have time a little bit for discussions and questions afterwards. And now we're going to hear from Dan, who has himself tried what Chihjen just taught us how to do. And it turns out it's not that simple sometimes.
That's right, I'm going to need your help. So as a recent hire at MFN, I'll explain it. Of the head of the tissue and DNA collection, I first started looking at NCBI's biocollection database in 2020, and the MFN was in there twice. One, here you can see on the far left, MFN, and it has the institution code ZMB, and a synonym, or alternative code ZMHU, when it was part of the Humboldt University, and several different collection codes. And it was also in there as MFN in the middle of the slide there, mostly for some mammal sequences. Now this is for institutions, the NCBI biocollections is institutions that have specimens that have sequence data in GenBank. And this is only genetic material. So I curated this, and now it's all only in there once, but it has the institution code ZMB and 2 synonyms, MFN and ZMHU. So pretty simple. But again, this is only genetic data, and I presented this at the Spinach and Tadwig meetings in 22 and thought, shouldn't there be a global registry? And thanks, Marie, you pointed out there is, yes, GRSciColl. So great for that. And we all know about this. So I started digging around in there, and not so simple. On the far left are search terms. I put in GBIF, and in the second column over are the different collection institution codes. Now this is mostly institution codes I'm talking about. The grayed out ones are redundant ones, so I put in these different search terms and just to exhaustively check how many times we were in there, and came up with a lot. And so going at it a different way, I went to GRSciColl directly and put in Germany and then Berlin geographically, and found about 18 museums. And there are a lot of museums in Berlin, but not that many natural history museums. So only about 2 or 3 actually. So 11 of these likely represent MFN. So a little history about the museum. It's pretty complicated and very old. And so it started out as the merger of 3 different museums, anatomical and zootomical. So this information I got from the wiki site and from the institution's homepage, but zoological museum and mineralogy. And each one of these is often considered different institutes. And these were all merged as the museum first officially formed as the Berlin University in 1810. And then on even then it had a few different names: Naturkunde Museum, Museum für Naturkunde, and Museum für Naturkunde Berlin. And then in 1949, it became part of the Humboldt University of Berlin, and that's where you see the ZMHB, HUM codes. And then in 2009, part of the Leibniz Association, and to present, it's called officially the Museum für Naturkunde Berlin, the Leibniz Institute for Evolution and Biodiversity Science, which I will just refer to as MFN for now. And it still has these three different institutes or museums: zoology, paleontology, and mineralogy. I'll just talk about the first two today because those are mostly relevant for this topic. So looking back at those unique codes that were retrieved from GRSciColl, there's about 20, but I could quickly eliminate a few of those that were in other cities around Germany. But 10 of these likely represent MFN for my initial inquiry. So I had some work cut out for me, still do. And so I'm not going to go through all these, just a few examples, obviously, because time. The first one, the MFN, the Museum für Naturkunde, the institution code MFN has about 771,000 specimens right now. This is also only specimen data, not observations and other types of entries in GBIF. About 627,000 of those are fossil specimens. About 138 material citations. I'll talk about those two in a minute. But as far as preserved specimens, there's about 143,000. And they have the collection codes. They have these collection codes you see on the bottom. And two of those that are in orange should actually be institution codes, not collection codes, MFN and ZMB. And those are the number of specimens that are currently in GBIF with those. The other ones, for the most part, are correct. Heteroptera, typically they use Diptera, they use the order as the collection code for the various collections. And so some you can see Diptera, 1,200 of those have ZMHU in the catalog number. So yeah, that's going to be complicated. ZMB, they're about 244, and I downloaded all of the data and then looked on the column of order, the taxonomic column, and found these. Most of these are bird orders and should all have the collection code AVs. Coleoptera, that could be the collection code, so that could be easily changed. Let's see, the fossil specimens, again, there's a total of 771,000 specimens, 627,000 are fossil. Now, 500,000 of those are from this Neptune database. And that's a database that was created at MFN, but it's occurrence records for microfossils from around the world, and very few of those are actually from MFN, so they should probably not be in there as specimens at MFN because they're not. About 127,000 of those fossils are not the Neptune database. So if we look closer at those, we see this column here in the middle are technically the collection codes for those 127,000. And these are basically upload files from MFN. So they're databases for all these various collections are very in the preliminary stages of being databased. And so we don't have an institution database that you can query. We have Specify, but very few collections are actually in there. People use Specify, Excel, Microsoft Access, various different ways to curate their data at this point. And these collection codes are basically the upload file names. And they all have the MB in the catalog number. So that's the institution code for these 127,000 fossil records. And so Paleo uses MB for the institution code, and then they use various collection codes depending on the organism. So in the middle, it's for vertebrates, and on the right, it's inverts. And so as an example, you see MBAM123, that's the Museum Berlin Amphibian Collection, and then an actual catalog number. And so these are in GBIF and GRSciColl under MFN institution code with these collection codes, MB.B3226. So MB, the institution, is in GRSciColl, but there are no specimens associated with it. So somehow I need to put this under the MFN umbrella with an alternative collection code, MB, and migrate these all over so they're properly curated. Okay, ZMB, the next example. 888,000 specimens, about 887,000 of those are preserved specimens with the collection code ZMB, which again should be the institution code. So about 1,000 of these are material citations, and all of those are from Plausi, which I'll explain in a minute, but pointing out some of those are actually AM&H specimens, SMF, which is in Frankfurt, USNM, so there's a lot of errors in that. Sorry, of these 887,000, most of them have these collection codes. We could probably drop the word collection, but for the most part, they're correct. Bryozoa, Nigeria, those are the collection codes they use, so those will be easy to fix. Only 155 have the collection code ZMB, which again is the institution code. So another example that was in the initial query is BSM, which was listed as the Berliner Städtisches Museum. And there are about 5 records in there, and they're all these material citations, and these are from Plausi. And Plausi is a nonprofit that promotes accessibility of data, and they use an automatic process that extracts information out of publications, scientific publications. So it looks for specimens cited, and then it grabs the catalog numbers and tries to populate them into GBIF. And clearly these are, if you see that red circle on the other side and at the top of the slide, these should be the Botanical Museum in Munich. So these have nothing to do with Berlin or MFN. And again, the Plausi has a lot of these kind of mistakes in it. So as a summary, these are the different codes, and it looks like there's about 8 of these actually do belong to MFN, 8 different institution codes, and each one of those is going to have several different or many collection codes associated with it. So there's a lot of work to do here, so I will need some help with this, but it's good to get a little preview of how I will do this. So in conclusion, for you at institutions, curating your institution, check the codes that are in GBIF and GRSciColl. Caution users of inaccurate numbers. So here, the 771,000 specimens might only be 500,000, or if the Neptune might be less than 200,000 actually that are in there. Proof automated systems such as Plausi, because this is ongoing, it's going to be continuously doing this. So I think we have to keep an eye on that and see if there's a way to fix it and get it more accurate. Verify these bulk added records like Neptune database, and going back and correcting a lot of the records that were initially recorded incorrectly is going to be a lot of work. So with that, I'd like to thank you for your attention, and thank you, Marie, and all the people at MFN that helped me.
Thank you, Dan. I hope everyone is scared enough now. No, thank you very much. So as you can see, sometimes what we present as straightforward is not as straightforward, especially if you have a very old, very big institution under your belts. We still kind of don't have really a lot of time for questions now, but afterwards, I'm going to be presenting how you can get involved in GRSciColl, and then we'll have time to discuss everything. If you have questions, if you need coffee, if you're regretting life choices right now. All right, I'll start with my presentation. So I just want to invite you to contribute to the development of GRSciColl, of the Global Registry of Scientific Collections, and I'll tell you how you can do it. And on this slide, I made this little hermit crab thing because I like hermit crabs, and somehow I feel like it fits GRSciColl. So, all right. So what makes the Global Registry of Scientific Collections? What makes GRSciColl? It's content and the user interface. And so I like the hermit crab because it kind of has content. It's GRSciColl. It's like what you see in it, the data. And that's what came to GBIF. And then we have the user interface, which is the shell, which, you know, can change, and you can grow mold and animals on it. And also you can change shells every now and then, like when GRSciColl came to GBIF from the Smithsonian. And then we also have standards and vocabularies that help us make the interface, the in-between, the getting in the shell. It helps a lot to standardize things and make sure it fits and becomes accessible. So how can you contribute to the content? Well, obviously, we already saw Chihjen's presentation that tells us how you can actually directly edit the content by making a suggestion. You can also become an editor or a mediator. If you have questions about that, I can explain a little bit more about the difference. But basically, it's someone who's responsible for either an institution or a country. Some of you are responsible for countries here. You can also help us organize hands-on sessions, especially if you're in a particular field and you know maybe a bunch of people are from collections. I know the Paleo group, Paleo Data Working Group has had one hands-on session, and it was really nice. I think it can be really helpful to be doing it by thematic. So if you're thinking of, you know, in your institution or in your like groups of like just having a little bit of a marathon of editing entries, then I'll be very grateful and very happy to support you with that. And also, another way that we like to get information in GRSciColl is by integrating other content or synchronizing with other content. So we didn't go on too many details on that, but we do get a lot of content from Indexa.Bioram. And so potentially we could get content from other sources. We just need to set it up. So if you know of national thematic and or national or thematic registries that have information about collections that you think could be a good source for GRSciColl, it would be really helpful if you could let me know. So such as Indexa.Bioram or iDigBio, but those are already in GRSciColl. So if you wanted to make a suggestion, you can use the suggestion system. And if you want to become an editor, organize hands-on session, or would like to help integrate another registry in GRSciColl or synchronize with another registry, just email me at scientific-collections@gbif.org, and I'll be very grateful. So now, how can you help us make the pretty shell pretty? You can become a translator. So we didn't mention it, but the GRSciColl website is actually multilingual, but so far we only have a Spanish version. We could potentially have other languages, but it would require an effort, concerted effort from a community of translators. We don't want to have someone, just one person do it. It's not sustainable. So if you would be interested in contributing, then I'll show you how to do that. Also helps if you can share use cases. If you're someone who's interested in collection information, but you cannot access what you would like from GRSciColl, maybe there are specific dashboards you would really like to see, maybe there are specific fields you would like to search on, it would be really helpful if you could share that with us. And of course, it's in a similar vein, it really helps us when we have actually concrete feedback, and it can help us prioritize development. It can help us, improve generally the website. We kind of really need the community telling us what is needed. So if you wanted to become a translator, you would have to do it in this system we call Crowdin. I can send you the link, or I can show you. I mean, on the website, on the GRSciColl website, there's a link to how to become a translator. And for the rest, you can email me at scientific-collections@gbif.org, and or you can join our mailing list. We also have regular calls, like 3 times a year, and we have a forum. And all of this can be found in this page on the GRSciColl website. It's called Get Involved, how to get involved. Yeah, all right. You can also contribute to improving the vocabularies we use for searching on some GRSciColl terms. And you can also contribute to the Latimer core standard by looking at Sharon's presentation and looking at all the links that she mentioned. So if you're interested in improving the vocabularies we use, you should try to email vocabularies@gbif.org, where my colleague Cecilia will pick up and see how you can help us change the vocabularies, maybe definitions, maybe have vocabulary translation as well. And yes, for Latimer core, Karen, sorry. I wanted to say Sharon, but I said Latimer core, so has this GitHub repo, and you can also look at their wiki and discussions. All right, so just a quick note on the recent GRSciColl development. You've seen a lot of what is GRSciColl now, and a lot of it's actually quite recent. It's only been since like a year or so. So that past year, we have reviewed the GRSciColl data schema. We have set up this structured collection descriptors. We have this forum, this mailing list, these regular calls, and we also have a new website. So all of that happened within the past year. We're very happy about it, yay. We also starting the last part of our roadmap, which is about institutional surveys. And I'll explain a bit what it is in case you're interested in giving me your input. I think it would be quite helpful if there were a couple of people interested in telling me what they think would be best, because we're really scoping this at the moment. So yes. So first, you've seen the collection descriptors we had. Right now, a lot of it is it's brand new. It's like only a few weeks old that we have it on our website. And only a few descriptors are indexed. So you can search data based on the scientific name of a collection, of a specimen in a collection. You can base it on the recorder's name, the collector's name. There are a few fields on the index, but we would like to know what's really important, what people want to see the collection indexed on. What do they want to look for when they're looking for their collection. Also, Sharon mentioned the MIDs. We generally have a challenge on how to work with quantitative descriptors because there's like a million of them. Everyone has their own quantitative descriptors. And you've seen Melissa's image where there was a table of descriptors that came from IndexaBioRom, and basically they have a number of specimen digitized, databased, a number of specimen imaged. I know, for example, that the National Registry of Colombia, they have all kinds of also custom metrics that have number of specimen identified at the phylum level, for example. And generally, I imagine all the institutions have their own metrics. And it's really, would be nice to be able to share them on GRSciColl, which is great, but it's impossible to index because we cannot index all the possible metrics in the world. So there is it needs some concerted effort, some definitions to know what really is important and then have that being in a standard living somewhere so that, can be used for indexing. Also right now, collection descriptors can be uploaded by people who have editor permission only, but maybe it should work with our suggestion system. We're thinking about it. And I put IPT ? So for those who are in the GBIF circles, maybe you know that you can publish data on GBIF via an IPT, which is a tool that helps you map data and publish it, mostly like digitized specimen type of data. And so much more. But, in our context, we're talking about digitized specimens. Could it be used to just share descriptors, like just higher level things like, like a group of specimens that are algae or, things like that. So that's also an open question. If you have strong feelings about this, just let me know, that would be great. And now, what I call institutional survey in our roadmap. So we've seen a lot of things, and maybe, at some point you phased out, and I understand, I would too, I do it sometimes. Let me bring you back in, because that's one where I really need input. So you might be aware, especially if you're part of institutions, that sometimes you send those surveys and you reply, and people want to know what you hold in your collection. So maybe you've designed a survey and you wanted to know what people had in their own collections, and so you fill in the form, and that goes back to maybe 11% of the people surveyed send back something that goes to a paper somewhere online and say, well, we have surveyed that many institutions and they have those in those collections. And each survey might have a different schema, different focus. You might have, a survey for pollinator collections, or a survey for staff capacity in institutions, and so on. And so they go online in a paper, and kind of, unless you're in those circles and you know that this survey was published, it's really difficult to access what is in there. And it's really difficult to know, well, so if I want to know about that institution, how do I know it? There's some dashboards online, but you have to know where to look for. It's not necessarily easy. And the idea for us would be that, wouldn't it be nice, and once again, it's an idea, we haven't actually done it, wouldn't it be nice if people who make those surveys could upload the output of the survey in GRSciColl? And then someone looking for a collection, they find an institution, and they're like, oh, they also replied to that survey in 2005. And thinking about it, if the survey is formatted, the output of the survey is formatted with some standard like Latimer core, it might even be something interpretable that could be displayed. And I'm not saying GRSciColl should be a place to make surveys. I'm saying it would be good to store surveys, results, not survey. But if all of a sudden you have all kinds of survey results available in one place, maybe it can also help people who design surveys to see what other surveys are out there on maybe similar topics. Maybe they can already gather some information that was previously gathered. It could be a place. It could be a place. That's the idea. And so for us to implement this, it would really help us to have more example of surveys of institutions that were done, and that would be nice to put in GRSciColl. I mean, I know of 2. I know there are many more out there, and so if yourself have been part of surveys, or if you've run surveys, it would be really helpful to have them. And another question that was raised at some point by people I was talking to was well, why stop there? Why not make GRSciColl a place to collect survey inputs? And why indeed? If the community would really want that, it would be nice to know. I mean, it could also just be so it's an open question. Another thing is, when you have those survey results, maybe a lot of them can be translated as descriptors that can be indexed. And so how can we do that? Is this something we want to spend effort on? Because it probably would be quite extensive effort spent there, but maybe it's worth it. So those are all open questions that, if you have ideas on that, I would really like to hear. Other ongoing conversations, you can join our forum and look at them and contribute there as well. Or should we mint DOIs for collections to be able to cite a collection, if you're in a paper? Also, now that we also have those collection descriptors with a bunch of recorders' name for collections, collector's name, maybe there's a better way to link collections to bionomia, for example, in order to give attribution, something more than just specimens on GBIF. If you don't have digitized records for a collection, but you do have a lot of collector's name, it might still be worth it. One last, it's a bit of gimmick really, but you can upload images to your GRSciColl pages, and we're wondering, like, for those institutions that don't have images but have a homepage, would it make sense to just pull from the website and display that? I mean, this is the type of questions we can have on the forum. So if you're thinking of any of those, you can go on the forum and just continue the conversation there. So I think that now I'm way past my time, but it's fine because it's my session. You're my captive audience. Also, it's the last talk, so So, so I want to feed the crab next year. I heard that hermit crabs actually can eat anything, and I was considering trying to draw a decaying octopus, but I thought it would be maybe a little bit too much, so here's a strawberry instead. So we want to continue adding content to GRSciColl, make the crab grow, right? And so for that, I would like to create material for helping uploading collection descriptors, organize hands-on editing sessions, and I would really like it if you're part of a community who wants to make collection visibles on GRSciColl, because I think that would be the best way to update information there. So that's the immediate next plan for next year. I think it would be good to focus on the content. Yeah. Thank you very much. Thank you for the speakers. Thank you for the GRSciColl team. We have developers that are super good, and yeah. Now, well, thank you very much. So for those who are tired, I won't hold it against you if you want to leave now. But for those who are motivated to have questions, we can have the questions from you. You can also help me answer the questions I just asked. And we can have a little bit of a discussion. If you prefer, opening your laptop and calling on one of our experts here to help you edit GRSciColl content, you're very welcome to go ahead and do so. So, yeah. Yes? Okay. Can we have online people being displayed? The Zoom on the... Zoom on the screen? Okay, you can read the question maybe. They wanted to talk. Oh, can we, the Zoom people, can we display it on the screen? That's fine, okay. Okay, we'll read the questions then.
We have 2 online questions from our online audience. And the first one is for Lily, and it is from Esteban. And the question is, do you have metrics of the success rate of contacting collections versus mobilized collections?
Thank you for the question. I think I had that displayed in the progress slide. So there were 19 institutions interested so far. So we are in the process of doing that, but so far 19 institutions were interested in mobilizing their collection, and it was 6 of them that's mobilized 161,000 specimens in GWIF so far. So I don't exactly remember, but we have that metrics and we can provide you later on if you are interested. Thank you.
I have a follow-up question related to that. For the examples that were on the screen, were there any metrics as to how much had already been mobilized for that country before these examples were done?
Actually, these are from all of our team, and I haven't been able to get in that detailed conversation with them. So I think there must be some, because SALSA was already working with the institution, Natural History Museum of Bulgaria, yeah. So there must be some specimens that were already mobilized. We might have some, but I'm not aware of that. So I think we can look into that too. Thank you.
Okay, the next question is for Marie. And there are two questions from David Fitzmuller. The first question is, are ROR IDs for institutions supported? From quickly sampling a couple of institutions, I was not able to find any. Are there any plans to import them? Connecting institutions with their ROR and Wikidata IDs, I have seen it very rarely, would really help matching with external data sources. And then the second question is also, is it possible to download the data as a dump?
Thank you, David. So I have used some tools to map ROR and Wikidata to GRSciColl, and I've imported all the identifier I could import by doing this automated mapping. So there is a bunch of ROR identifiers and Wikidata identifiers. It's still a small percentage of institution entries with those, but they're the ones that could be mapped automatically. And also, institutions, collections are very welcome to add those identifiers themselves to the entries. So anything that's not automated, that wasn't done automatically at that point in time, they can still be added manually. But it's still a relatively small percentage, and it's because it might be sometimes difficult to identify, I think, the names when you have them in different languages and so on. So it's difficult to automate. But so that was my attempt. And if anyone has a good idea on how to do it, otherwise I'm happy to also use that and import more identifiers. And the other question is, yes, you can totally get a dump of a GRSciColl search result. So it could be everything or just a part of it. If you go to the GRSciColl website, whether it's an institution search or a collection search, at the very top of the table, you have a button that says download as TSV. Just between the search filters and the table, you have like a little line. It's a clickable link. And whatever you search, you can download and have a TSV file out of it. There's also an API call if you prefer. But then it gives you a whole table with your result. It might be sometimes easier when you want to check a really big selection. So hope you answer the questions. Yeah. Okay. Do you have any question from the audience here? Yeah, yes. Please, just one after the other.
Sorry, I don't know who I've been. Javier Molina from ALA, and thanks for the great presentation for everybody. My question has to do with whether there is a similar concept on GRSciColl like the JBIF hosted portals for observations or occurrences, where you can customize the views to show a particular subset of records, like per region or country.
Yes. So actually, GRSciColl content can be shown in hosted portals, and in fact, the current GRSciColl website is a hosted portal. So and can be for a subset of, it could be, you could have a hosted portal for a bunch of thematic collections, right? So if the data is there and if it's full enough for a selection, then it could be made into a hosted portal with all the data from GRSciColl. Yeah. Yes, please go ahead.
Hi, Alina Freire from Ecuador. I remember years back when we started working a little, we had a question whether the information from the index herbariorum is fed back and forth. Has that been already established or not?
No. Right now, it's only updates in index herbariorum trickles to GRSciColl, but not the other way around. So there is no way to send back information to index herbariorum. But it's a question we've had several times more, and so I think we'll have to talk with index herbariorum, see what would be possible, but right now it's not the case. Yes, please, Nikki.
Yeah, It's to do with when you log in to propose a change. Is that your GBIF login that is used, the GBIF data portal login?
Yes. So if you want to become an editor and have permissions for your editing, then you need to have a GBIF account, yes.
Okay. I just wondered if you were doing identity management that wasn't through the GBIF data portal, if that was an opportunity to get more people to get an ORCID, if you used ORCID logins for that.
Well, I mean, if you can log in the GBIF portal with your ORCID,
Yeah,
so yeah.
Well, yeah, I know that now.
Yeah, no, exactly.
And the other thing was the call for use cases that you had in your talk was to an email address. And I think Sharon's talk was really nice about how the development of Latimer core and stuff was all done on GBIF in the open. And I think people get inspired if they can read other people's use cases to think about different ways they can use a resource. So if that could be done in the open too, I think that would be a positive thing.
You mean make it public?
Yeah, your request for
my request.
use cases was just to an email address.
Yes.
Whereas if others can see what other people's use cases are,
then
that can be quite inspiring and that can help them think of ways that they could use it themselves.
Good point. Yeah. Actually, yeah, okay. Yeah, I'll think about it. Yeah. Thank you. Yes, please. I think Rod raised his hand first.
So this relates, I guess, partly to Dan's talk. So I just want to make sure I understood. You're talking about cleaning up the data. Was that in GRSciColl, like the codes and things? Or is that potentially going to be the collection? I guess my concern is sometimes museums decide to change institutional codes. Like GRSciColl, when it originally started, was partly trying to clean it up. Like the Natural History Museum in London has changed from BMNH, whatever it was, to NHM UK. One catastrophic implication from my sort of work is all these historical records in the old literature are to the old codes. So I guess my concern is this project going to essentially mean that people will change institutional codes, which means you get disconnected from the citations to your literature?
Yeah, that's a great question. And I think those codes, if they are in ancient publications, they need to persist. So I think this concept of having alternative codes that needs to be curated, and a lot of those things need to be fixed, I said, but they still need to persist. So what I'm envisioning is there's going to be an MFN institution in GRSciColl, and it's going to have a lot of alternative codes. It has to capture all those, because we don't want to get rid of them, because if there's a publication that uses these codes, and some of the challenges in that museum is that because it's not all digitized, some of the collection curators and staff don't even know until they go open those drawers, and so for instance with the Lepidopterans I found through Plausi, some had ZMHU, and I asked the current curator, and he uses MFN LEP for Lepidopterans. But there are some, and he went and checked, because I found some that were added through Plausi from a publication, and he was like, yeah, they have ZMHU as the collection code, so that needs to persist. Yeah. Yeah, another.
So I just wanted to comment a bit on this point. So there are also countries where you have several acronyms per institution because they are multilingual. And actually in Disco we use the ROR ID, which is very handy, because similarly as we do with synonymy for species, you can add all the current and old acronyms or names that were used, and the ROR ID interlinks them, and so that you can, when you find an old name, what it's linked to the current institution where the ROR ID is the valid ID. And I would also like to highlight the strength of Plausi in this, because with the text and data mining, they can find and uniquely identify all these old names, being it acronym, species, and so on. And of course, there are mistakes, misspelling, and so on, but it's not to mistake the usage of that, because of this work they are doing, even if there are errors, it gives us the possibility to do these synonymies. So it's not because there are errors that it's not good. So we have, of course, works to do, but they have the merit to find all this
Yeah,
old
yeah.
and mistake name, and that we can correct them.
Sure. Yeah, I'm not advocating getting rid of Plausi, just to be on the record. But yeah, nobody's going to go back and look at all that old literature, but AI will, with the automated software, we can do that. Yeah.
Unless you want to add something more.
No, I was just going to say one to the previous question, just thinking about this, and I always think about genetic information, and a lot of people, I know, put things in GenBank and they have a catalog number that's a collector number, but then that collector number is in the publication. And so I've always, myself, have come across, I've done this myself because the specimens weren't yet cataloged. And GenBank has other fields. They have an isolate field. And so what I've done later is gone back and changed the records. People don't also realize they can do this. And they say, oh, it's in GenBank, it's already done. Well, you can change it. But just be careful. And so I would put in the correct voucher number for the institution and then move the collector number that's in the publication to the isolate field. So it's still in there in the record. So it's still tied to that publication. I've done this with a few of my own, and you can only change your own. So you have to be one of the original submitters to change it, is the only. Yeah.
Yes, we have first, yes. Yeah, he was trying like 2 people ago. So, yeah.
Well, I'm not familiar with the GRSciColl, but I have 2 questions. One is a simple one, and librarian people have a standard system of called ISIL, International Standard for, I'm not sure, but that is a code for a library, as it's called. But they expanded to the museums sometimes. In the case of Japan, well, the National Diet Library people wants to put the number to any museums, so that, well, do you have such kind of interoperability with the ISIL people or with GRSciColl? No?
No. We don't have anything directly with ISO, but it's maybe something to explore, actually. I haven't looked into it. Maybe that's a very good idea. I wasn't aware that they were actually working with museum data as well.
I
So,
see. Yeah, well, why I am talking about that sometimes, well, such as Europeana or Japan Search, such kind of any kind of cultural resource system, they want to be based on the ISIL, or so sometimes such kind of well, Japanese GBIF is connected with our Japan Search, so that such kind of interoperability would be needed in the future, I suppose.
but maybe in the microphone, yeah.
Okay.
Thank you very much.
So, because
No, but you
TEDWIC
can-
is about standards, I mean, the executive, of course, we are in contact with ISO, and we were very much in contact for everything that is DNA ISO standards. The problem is that ISO standards are, one, not open for many. They cost a lot. You have to pay national registration. You have to pay extra to be in all the committees. So it's not possible for TEDWIC to collaborate directly, but we have exchanges about the content and the data. And our colleagues from ISO, they made another site that is not ISO accredited, but where you can still have access to the information. But for our community to remain true to openness and fairness, ISO is difficult. And we do it only when we need an accreditation by the EU Commission, because they force us to do ISO. But for the rest, we prefer to remain open and that people don't have to pay to use the standards.
Okay, thanks. You had a follow-up question?
So just to quickly clarify, I don't think we're talking about ISO.
ICO.
We're talking about ISIL.
ISIO. Okay. Oh.
This is it's a Japanese system, and again, this is only discovered that through Wikidata, which I would argue, and Siobhan's probably desperate to argue, solves all these problems using linked to it. So you could link to the Japanese, I'm just looking at Japanese.
Yes,
Wikipedia.
yeah, okay. I need
So I
to come
thought that,
afterwards and grab the URL.
There's
Yeah.
so many acronyms flying around.
Okay, thank you, sorry.
I don't know that.
Thank you, thank you.
Okay. I want to ask another question. I think there is a possibility to utilize GRSciColl catalog series. Well, there's a lot of undescribed collections in a small museum, especially, and there is no database well, but they should appear what kind of collection they have, so that at least you mentioned that at least you need one record for to make a catalog data.
Yeah.
So that is. Well, unless they do the one data to GBIF, you cannot make a catalog data output.
No, you can. GRSciColl, you don't need to publish digitized specimens in order to be on GRSciColl. You can even have personal collections in GRSciColl. If you have your own personal collection that you would like to make discoverable to the world and say, I have a collection of, Coleoptera. It's a small collection, but it's from Coleopteras of Japan or something. You can register it yourself, and people can find it that are interested in the content you have. And it doesn't have to be on GBIF ever. It can be, and doesn't have to be digitized. And that's kind of one of the points of GRSciColl, because not everyone can digitize everything.
I see.
So it's fine to just let us know it exists already. It's better than not knowing it exists.
Yeah.
So if you actually know of small collections that are invisible currently, we can spend some time adding them, making sure that they become more visible. And it doesn't have to take that long. I mean, Dan's case is like really big institution, right? It can be much faster to do sometimes.
Thank you.
So yeah. Please, maybe we can give you the microphone otherwise. No.
Yeah, sorry, I was just following up a bit on Rod's questions, Elspeth Haston from the Royal Botanic Garden, Edinburgh. I was just thinking the relationship between Latimer Core and GRSciColl, and the idea of being able to implement Latimer Core fully in GRSciColl could allow us to retain that connection between some of the historical collections and the current collection that is held within. So, for example, we've got a lot of specimens from the Glasgow Herbarium, but we could actually make that connection between the individual specimens and the original herbarium collection that it was in, although it's now housed within the Edinburgh Herbarium. Is that something that will be functional?
So right now, I'm going to talk about right now what we have for this type of situation. So you could, of course, make one entry for a historical collection that no longer is active, but was active at some point, because, for example, you might want to showcase that collection has a historical value. Maybe it burned down. It's like not active because it's just not physically there, but you still want a record of it. So that you can create like one GRSciColl entry for it, just say it's not active, explain the situation, and so on. But with the collection descriptors, you could also consider doing something where you have your main collection as one entry, and then you have maybe one table per sub-collection with some descriptors associated with them, or you could have one big table and one row is a sub-collection, depending on how you would like to describe it. So because this implementation is so new and Latimer Core is so flexible, we don't have yet guidelines on how to best do that. But if you have a use case, that would really help us actually make the guidelines. So yeah, you're very welcome to come and then we can do it. Yeah. Well, thank you very much, everyone. Hope that was somewhat insightful. And let me know, well, like we're here during the conference, we can help you. You can come to the GBIF help desk. And yeah, I'm very happy you all came. Thank you very much. If If you have further question, now it's a bit bad. There's also Oxford Abstract Virtual if you want to continue the conversation.